No mai, haere mai. Welcome everybody to tonight's webinar. My name is Nicola Batossi and I am CEO of Fertility New Zealand. It's my absolute pleasure to welcome you here tonight. This is the last of our Lockdown Wednesday webinar series. We will continue to hold webinars, but about once a month going forward. So thank you to everybody who's joined us over the last 6 or 7 weeks. If you've missed any of them, you can just go to the news section of our website, fertilitynz.org.nz, and all the recordings are available there. So Fertility New Zealand has been supporting Kiwis with fertility issues for 30 years now. We operate nationally. We've got a team of wonderful volunteers that run support groups and events around the country. We have a range of information available through fertility clinics and on our website, and we also represent the voice of New Zealanders with fertility issues. So if you're not a member of Fertility New Zealand, please join us. That supports all the work that we do, lobbying and other advocacy work. Every member that we have makes our voice stronger when we're advocating for change. And please do contact our helpline, which is free of charge, completely impartial, and would be very happy to support you in any way that we can through your journey. So without further ado, it's my absolute pleasure to introduce tonight Jeanette MacKenzie. She is Scientific Manager at Fertility Plus Clinic in Auckland. She's got a breadth of experience in the field of embryology. Prior to this position at Fertility Plus, she was scientific manager of a different fertility clinic in the UK. So she's got New Zealand and international experience in the field. So I will now hand over to Jeanette. If you have any questions, Jeanette will be very happy to answer them at the end. Feel free to type them into the box, but we probably won't approve them until the end of the presentation for her to answer then. So thank you very much, and over to you, Jeanette.
Hi everyone, I'm so pleased to be here to be able to share my amazing job with you all. So I'm going to be talking about what an embryologist does and what happens in the laboratory. So, and some of the techniques that we use in assisted reproductive technologies, mainly IVF and ICSI in the lab. So you may not have met an embryologist before and know what we do. So we're basically the ones behind the scenes looking after your eggs and sperm and embryos. We, in New Zealand, we're all specifically trained and we are all registered to the Medical Laboratory Science Board here in New Zealand. And that means we've got annual competencies and CPD to follow. So yeah, that's a brief outline of what an embryologist is. So let's get on with the talk. Where did IVF all begin? Well, it's over 40 years ago now when the first IVF baby was born, Louise Brown, and she was born in from the clinic of Bourne Hall in England. And this was Sir Robert Edwards and Patrick Steptoe, and they had been working for years and done so many experiments and trying to get IVF to work, and finally it worked after probably hundreds of attempts at fertilization in the lab, and Louise Brown was born. It was quite controversial at the time. As you could imagine, in 1978, this baby being born from this new science fiction thing called IVF. Then in 1980, Australia followed, and the first Australian baby was born, and that was Candice. And then in New Zealand, a few years later, in 1984, our first baby, Amelia, was born. Back in those days, success rates were really low compared to what we expect these days. and on average it was about 10% chance in the early 80s. When Louise Brown's mother was trying for a baby, she was told by the doctors that her chances were 1 in a million, and she got that 1 in a million baby. So a typical IVF process involves stimulating the ovaries with hormones, involves collecting some sperm, it involves collecting the eggs, it involves embryo culture, which means growing the embryos in the laboratory, it involves a transfer of the embryo to the uterus, and then cryopreservation, which is the freezing of embryos. So we'll go into some more detail about some of these processes. We're going to skip the hormonal stimulation and get to the lab once the eggs are collected. So this is a bit of a cartoon about what happens in the lab. This represents an egg, and this is on the day of egg collection, which we call day 0. And then we also have the sperm collection on that day. So egg and sperm is collected on day 0. With conventional IVF, the sperm is washed in the laboratory, and then thousands of those washed sperm are added to the eggs in a dish, and those sperm are just left overnight in the dish to find their own way to the egg and fertilize overnight. And then in the next morning, the embryologists will come in and check these dishes and see which of the eggs have fertilized. And that's what we call a 2PN embryo. And that means 2 pronucleate, because in a fertilized egg, there's 2 little circles in the middle, and they're pronuclei, and they represent the chromosomes from the sperm and the egg. So the day after an egg collection, the embryologist will ring you as a patient and let you know how many of your eggs are fertilized. That's what we're talking about at that stage, is a 2PN embryo. And that's day 1 of the embryo in the lab. In the afternoon of day 1, we expect the egg to start dividing into the embryo itself. And we usually see in the afternoon or into the night it divide into 2 cells. So if you can see where I'm pointing here, this is representing a 2-cell embryo, and that's usually the afternoon after your fertilization check. The next day is day 2 in the laboratory. And usually on this day, we see something around the 4-cell stage. Sometimes we don't look at the embryos on this day, but sometimes we do, just to see what they're doing. And then the next day is day 3. And on day 3, we expect to see something around about 8 cells. And it doesn't have to be exactly an 8-cell embryo. It could be 6 or 7, 8, 9, 10. And by this stage, embryos are starting to look different from each other. And in some clinics, we would consider doing a transfer on day 3 if we can clearly see which is the best embryo. But quite often, all the embryos in the dish look quite similar from each other. So quite often we actually go on for 2 more days to day 5. And this is the stage where we call the embryo the blastocyst. Now, you can see in the cartoon how it actually looks like a completely different structure to a day 3 embryo, because in those 2 days, from day 3 to day 5, the cells are starting to change and differentiate. So that means they're starting to become certain kinds of cells that is going to develop in the embryo. And in a blastocyst, you can see there's a clump of cells, and that's called the inner cell mass. The inner cell mass will go on and form the actual fetus, the baby itself. And then there's cells that migrate to the outside of the embryo. And around the outside are all the cells that will become things like the placenta and the sac. So, yeah, the blastocyst has done a lot of work in the lab to get to that stage. Now, we'll just skip to the next slide, which is exactly the same, but it's just with ICSI. So we routinely do IVF, the standard insemination, in anybody that comes for IVF where the sperm is normal. But if there's any worries about the quality of semen, so it might be low motility, or it could be a low count, or maybe the way the sperm are swimming or the way they look like. And if the embryologist is not quite happy with the sperm, that's when we recommend an alternative to adding the sperm and egg together. And this is what's known as ICSI, and you would have seen this all over the news and media, because whenever they talk about IVF, they always put a picture of ICSI, which is down here, an egg with a needle going into it. So when the sperm count is low, we like to give the sperm a helping hand to get over the barrier of swimming to the egg itself and being able to get inside the egg. So the embryologist will use the washed sperm, and that's washed in the same way as for IVF. And instead of just mixing thousands of sperm and with the eggs, we would actually take one sperm inside an injection pipette and carefully put the sperm inside the egg. And that way we know for sure the sperm has got inside the egg. And that's what the ICSI process is. Now, from that moment, everything is exactly the same in a cycle of IVF. So if you're having ICSI, it's just the way the sperm gets inside the egg is different. So from there, we do the same thing in the morning. We check for fertilization, ring you to let you know how many eggs are fertilized, and then the embryos will be growing on in the same way. Nothing is different for ICSI with the embryo culture. After the culture happens and we do the embryo transfer, in New Zealand, majority of embryo transfers are single embryo transfers, and New Zealand has been quite a leader in the world about doing single embryo transfer to minimise the multiple pregnancy rate. And so we've been really quite good in New Zealand at lowering the twinning rate, which is a great thing because we know that multiple pregnancies are associated with poorer outcomes. After an embryo transfer, if any of the extra embryos that are left in the dish are also good quality, then they can be frozen. Not every single couple that comes through for IVF will be lucky enough to have frozen embryos, but a good majority of people are. And if you do get frozen embryos, that means you'll be able to either store them for siblings if you're successful on the first cycle, or if it doesn't work with the fresh embryo, then you can try with the frozen embryo, and that means no egg collection for trying with your frozen embryo. So the next part of my talk is about extra things that we can do in the laboratory, and these are known as add-ons. And most of these things can be done if you pay for them. So I'm just going to go through some of the ones that we are offering in New Zealand and talk about some future developments as well. So you may have heard about this one, PGTA, which is pre-implantation genetic testing for aneuploidy. Now, aneuploidy is talking about the number of chromosomes in an embryo. So by doing this test, we can check blastocysts, and this is a picture of a blastocyst down here. We can check if the blastocyst has got the right number of chromosomes in it. And if it doesn't have the right number of chromosomes, if there's an abnormal number, it means that that embryo has probably got less of a chance of developing into a baby. So the embryologist will do a biopsy of the embryo, and this means removing a few cells, and it's usually between 3 to 5 cells. And those cells are put in a little tube and labelled with the number of the embryo, and then the cells are sent off to a genetics lab to be tested to analyse what the chromosomes are like. In the meantime, the embryo is frozen and stored in your laboratory, in your IVF clinic, while the cells are sent to the other genetics lab. And then we wait for the results, and it usually takes about 2 to 3 weeks for the genetics lab to run the test and come back to us with the results. And then the results will say if it's got a normal or abnormal number of chromosomes. So if it's got a normal number of chromosomes, we would be able to choose that embryo first for embryo transfer. And if it's got an abnormal number, then we would that would be a low priority. So doing this testing is probably really only useful if you've got at least 2 to 3 blastocysts, because if you've got one blastocyst, you're probably not going to gain any information. So what the literature is saying is that if you do have 2 or more blastocysts, it might increase your pregnancy chance per embryo transfer, which means it might quicken the time to pregnancy. If you've got a few embryos and we transfer them in the traditional way of not testing, and say you've got 5 blastocysts, that might take 5 months to find the one with a normal number of chromosomes if we were unlucky enough to pick the normal one as number 5. But if we've done this test and we know that actually embryo number 5's got the normal number of chromosomes, then we would actually put that one back first. So it's an embryo selection tool. So therefore, this might quicken the time to pregnancy. However, overall, from an egg collection, it doesn't increase the live birth rate because it's not changing the chromosomes of the embryo. It's just telling us what the chromosomes are in each embryo. So from one egg collection, there's no higher chance of actually taking home a baby at the end. And there's a couple of things that we need to consider when we're doing things like this to embryos. Is the genetic test that is done is not 100% accurate, which means we always recommend some extra antenatal testing as well. As you can see from the picture, this is an invasive procedure. So we cut a little hole in the side of the embryo. It's only a cut in the shell of the embryo, so that's no bother to the embryo, but we are removing some cells from the embryo, and it seems to be that this is not affecting the embryo's ability to implant, but we are not really sure of the long-term growth on the embryo and therefore in the womb when the baby's growing, if that's going to alter any changes to the baby themselves. But there's more research to be done about that. The other thing which is comes about with this testing is that some of the cells that get taken from the part of the embryo might not actually be the same as the remaining of the embryo, because not all cells might be identical in their chromosomes. And when cells have different number of chromosomes in themselves, it's known as a mosaic embryo. So it might be that we've taken a portion of the embryo which says in those cells that there's an abnormality, but actually the rest of the embryo is normal and vice versa. So what we do with results like that is they would be our lower priority for transfer, and then we would refer to genetic counselling to discuss the implications of mosaic embryos, because they can be transferred. Just need to have the implications discussed with you. Now, in New Zealand, there's no public funding for the screening of embryos. So it's roughly at least $2,000 extra on top of a IVF cycle. So the next thing coming up, this is IMSI, and this is intracytoplasmic morphologically selected sperm injection. And this is an extra tool an embryologist can use while they're doing ICSI to be able to select the sperm. It uses a very high magnification, 600 times than the normal magnification that we actually use to choose sperm when we're doing ICSI. And what this means is the embryologist can see the sperm really close up and look at the head of the sperm. Now, the head of the sperm is where the DNA is stored, so it's a really important part of the sperm. And if there's any abnormalities, like in the picture on the left, you can see some things that look like sort of craters on the moon or holes. Now, that's not a good sign. So if we can see some things like that, then we wouldn't want to choose that sperm. Now, there's no extra risks to doing this process. It's just the same as doing an ICSI, so it's not the same risks as ICSI. And the research is showing that this might be useful in people that have had previous failed ICSI attempts. But Overall, the research doesn't show an increase in live birth rate for infertile men when we use this in addition. And it does have an extra cost on top of the IVF and ICSI part of the cycle. Another sperm selection technique is called PICSI. We love acronyms in IVF. So this one is the physiological intracytoplasmic sperm injection. And with this one, it is because research has shown that sperm that bind to hyaluronic acid, and that is a natural compound found in the body, but the experiments have shown that if these sperm bind to this hyaluronic acid, they're probably going to be really good mature sperm with lower rates of DNA fragmentation. So, you know, DNA fragmentation we can't see down the microscope, but this is a way of trying to select sperm with low DNA fragmentation. And DNA fragmentation is kind of what it means is that the DNA is fragmented, so not good quality. So to do this, there's just a different dish that the embryologists use, and the sperm are placed in the dish, and there's a cartoon of it there, and there's droplets of hyaluronic acid in the dish, and the sperm that swim to those drops of hyaluronic acid and bind to it, they are the ones that are mature and the best quality. So we will pick those sperm and inject them into the eggs. There's no extra risks for this process. It's the same as doing a normal ICSI. But unfortunately, after all the great research that's been done, a recent large randomized study hasn't shown that PICSI increases the chance of having a baby. And this is usually an extra cost as well. Not as much as IMSI for this one, but a little bit of an extra cost for that one. Another thing you've probably heard about is embryo glue. And this is a special culture medium that we use in the lab, and we put the embryos in it before embryo transfer. And it's got a very high concentration of hyaluronin and recombinant albumin. And these are really big macromolecules, so they're big molecules that are involved in promoting implantation. So when we put the embryo in this, it's kind of like a nice protective layer around the embryo and it's put into the catheter with the embryo, then into your uterus. So it's got a lovely, nice, safe environment to be transferred in. And then those lovely macromolecules are supposed to promote implantation. There's no risks to having embryo glue, or no known risks. And good news is there has been a big review of the literature, and it does seem that embryo glue can increase the pregnancy rate and the live birth rate, so the chance of having a baby, by about 10%. So that is great. And when clinics use it, and I'm pretty sure every clinic in New Zealand uses it. I mean, I don't work at every clinic, but I'm pretty sure everyone does use it for all their cycles. And I think this, the cost of it is included in the normal standard IVF cost. Yeah, so you can check with your individual clinic if you don't know if your clinic does it. Another media that you may have heard of, so this is one called EmbryoGen and BlastGen. Now, the two different ones are just for different stages of development. The EmbryoGen is for the early stage embryo, and BlastGen is for when the embryo turns into a blastocyst. And this is a media produced by a company, and it contains a growth factor, and that's GM-CSF. This specific growth factor is involved in cell division and growth and differentiation and suppression of cell death, so stopping cells dying. If you had this media, it would be instead of your normal culture media that the lab would use every day for everyone. So it's an alternative, and it's usually got an additional cost because we don't use it routinely. There's a few reviews about this, and It's not clear, there's no strong evidence yet whether or not this is actually increasing the rate of taking home a baby at the end of your cycle. So, other options we have got in the laboratory is time-lapse imaging, and these are special incubators, and these incubators have got cameras inside. So these cameras take photos of the embryos developing, and they're taking photos every sort of 5 to 10 minutes. And when we speed all those photos up, we get a video of the embryo developing. It's a really exciting thing for embryologists. And what's really good about it is we can put the embryos in the incubator and we don't have to even take them out to check their development or anything. We just put them in, leave them there in a completely undisturbed environment. That's really good because we don't want to be taking embryos out of the incubator too often, because that causes stress on the embryos when we're altering the temperature and the pH. So by just leaving them in there, we're giving them a really good environment. All the time-lapse incubators, you would have heard about probably EmbryoScope is one. The one in the picture is Jerry, and that's produced in Australia, that one. They're very high-quality incubators. So they're really, really nice incubators. And that one in the picture is basically 6 separate incubators. So each patient has their own compartment in the incubator with their own camera and their own temperature control and their own gas. So it's like having your own individual incubator. Now, why this is so exciting for embryologists is because we can see the embryo developing. Everything it does, we can see. We can see every cell division, and sometimes we see really unusual things that we've never seen before when we've just looked down a microscope at a moment in time. So what that means is if we see the embryo do something a bit funny, like for example, sometimes one cell might go directly into 3 cells, and they shouldn't do that. But if we see that, then we know that embryo's got a very low potential. So it helps us to select the embryos. And in this way, it's kind of like a deselection. We can see that that embryo's done something a bit unusual. We know it's going to have a low implantation rate. So we can put that as a very low priority for embryo transfer. The other cool thing about time-lapse is you as a patient can get a video of your embryo that you have transferred, which is quite a nice little memento to have. Research around the world where looking at developing algorithms, and this is looking at points in the embryo's development and giving it a bit of a score, and then we're able to rank the embryos depending on when the embryo did certain key points in its life. So when it became a 2-cell, when it became a blastocyst, and we can time that exactly within 5 minutes of when it does it. And then there's algorithms being developed to help us make even a better selection for embryo transfer, to get the best embryo, to get the best chance of pregnancy for your first transfer. However, we still need lots more research about this, because so far the research is showing that there isn't an increase in your overall chance of taking home a baby from one egg collection. But it's a very promising technique and very exciting in our field. It does cost. Usually it does cost extra to have your embryos in time-lapse, but check with your clinic, and not all clinics in New Zealand have got time-lapse either. So what about other developments and future options? We've got in vitro maturation, IVM, and this is available. It's just not very common at the moment. And this is an alternative to standard IVF. So in standard IVF, you have lots of injections and drugs. But this method is very, very reduced number of injections and hormones, and this might be suitable for women at risk of OHSS, which is hyperstimulation syndrome, or if oncology patients are coming through to freeze their eggs before their chemotherapy, and if they're estrogen receptor positive, we don't want to be pumping them full of more drugs because that might make their cancer worse. So IVM might be an option for them. So the difference is we don't collect mature eggs. We don't give the trigger injection at the end to mature eggs. We take the eggs out before when they're immature, and then the eggs are matured in the lab. So an egg collection will be done one day, and then the next day the eggs that are mature can be have sperm added. And this technique, we have to use ICSI for that. So it's a bit different to normal IVF, because on normal IVF, you have sperm and eggs on the same day. But this is different because the husband would need to come in the next day to do the sperm sample. Now, success rates are lower in this technique, and that's because usually there's much fewer eggs collected. And also this treatment is not done very often, so we don't have much information available on the long-term safety of this technique. I was looking on the internet at numbers of cycles done, and I could see in the UK about 15 cycles were done over 2 years. So it's really not very mainstream as yet, but it's available. Another new exciting thing that might be coming in the future is non-invasive PGTA. So remember, PGTA was testing the embryos for the number of chromosomes, and that's when we do the biopsy, it's invasive. But this way is non-invasive, which is meaning that we are not going to have to biopsy the embryo. Instead of biopsying the embryo, we take away the culture media that the embryo's been growing in, and look at the chromosomes that have and the DNA that's been secreted into the media. So there's a lot of research going on into this at the moment, and one small pilot study in Spain has shown that they got 84% concordance with biopsy results, which means they had 84% of the results were the same in the culture media compared to a biopsy. Now, obviously, that's not 100% yet, but it's a promising start. However, lots of other labs that have been in research trying to mimic this are not quite getting that same good results. So it is really limited by technical difficulties at the moment. But there is a large randomized controlled study underway around the world at the moment. So we are keeping our eye on that. We also, you may have heard of endometrial receptivity analysis, ERA, and this is available. It's when you have a biopsy of the endometrium to check about your window of implantation. So what the test does is look at lots and lots of genes that are expressed in the endometrium, and they can tell if your endometrium is at the receptive stage, a good time to receive the embryo, or if it's pre-receptive or post-receptive. And a biopsy is taken. It's sent away overseas. I think in New Zealand, our closest lab is Japan. It's sent away. And then the test results are sent back, and it says when your perfect window of implantation is. So, you know, we usually do transfer on day 5. They might say, well, actually, this lady's better to have a day 6 transfer or a day 4 transfer. So I know some of the clinics in New Zealand are having experience with this, and sometimes they are being told, maybe 1 out of 4, that they do need to change the day of transfer. So they change it, and some people are getting pregnant, but not everybody's getting pregnant. So it's not going to be the answer for all implantation failure. So at the moment, we say it just might be useful for people with recurrent implantation failure, but we need to do lots more research into this to find out really what the benefits are. And the last technique that I'm going to talk about is about the endometrial microbiome. So that's the healthy bacteria inside your uterus, known as the endometrial flora. So research has shown that there might be an association with the bacteria in the uterus and implantation. So in particular, researchers found that low levels of lactobacillus were associated with poor implantation. So again, this is a potential test, involves a biopsy. And again, may be useful for women with recurrent implantation failure. So that's like transfer of good embryo after good embryo after good embryo that's not working. And the results of the biopsy will come back from the testing lab, and they may recommend a treatment of probiotics to enhance the healthy bacteria in the uterus, to enhance implantation. This test isn't available yet, and we don't have any studies yet to confirm if there's an increase in live birth rate, but it's really encouraging to see this kind of research being done, and exciting that new things are on the horizon. And that is my talk finished, so feel free to ask any questions. Okay, yep. How are embryos stored? Well, they when we freeze them, we need to take all the water out of the embryos, because if we freeze the embryos with their intracellular water still inside, it'll form ice, and ice is going to kill cells. So we take out the water by dehydrating them with a kind of antifreeze, not the stuff that you stick in your car, but a special IVF one. And we put the cryoprotectant in the embryo, and then we store it at a very cold temperature, minus 196 degrees. It's in liquid nitrogen, and they're stored on a little plastic half a straw thing. Yeah, it's on a straw with a cap on the top, and all labelled up with your name and date of birth and NHI. And how long do they last when frozen? So kind of forever, really. Once they're under the liquid nitrogen and at that temperature, they just stay like that until you bring them out. There's been I know there's been sperm thawed and used, like 20 years old sperm has produced babies and embryos. So, but as you know, the law in New Zealand is store for 10 years, and if you want to store for longer than 10 years, you apply to the ethics committee. No, 7 cells is absolutely perfect for day 3. Something around 6, 7, 8, 9 cells. A 7 cell just means that that final cell hasn't quite done its division to get to 8 cells. Natural IVF is different to IVM. Natural IVF is where we just monitor the growth of one follicle. Sometimes you have a little bit of clomiphene, and then you might get, say, 2 follicles. So IVM is different to that. So natural IVF, no, doesn't usually use injections. It's usually just clomiphene. So yes, it's definitely worth to transfer a 7-cell embryo.
Thank you very much, Jeanette. It looks like that's all there is with regards questions, unless anybody has another question that that you'd like to post in the box now. Oh, I have a question. Okay. If you decide to post that question pretty quickly, and then we can wrap up. Thanks, Jeanette.
No, you can do IVF to have PGS. We used to have to do ICSI because of the genetics lab and the techniques they were using in the genetics lab. But these days, no, we can use IVF because they've developed techniques not to let the sperm DNA interfere with the tests.
Thank you so much, Jeanette. Thank you for sharing your all your expertise with us tonight. It's been really valuable, and I hope that everybody who's attended has learnt something about this fascinating and rapidly evolving science. So thank you very much, Jeanette, for joining us tonight from Fertility Plus, and to everybody as well who has spent their Wednesday evening with us. So we look forward to supporting you in the future, and thank you, everybody, and good night.